so let's get started. Welcome to our weekly seminar. I'm playing the role of Todd today. He's on an airplane headed to Southern California. I want to introduce Dr. Matt Roby. Matt comes to us from the USDA-ARS in Davis. That's the Agricultural Research Service. It's the research arm of the USDA. Matt is from California, from Brentwood, California, kind of at the foot of Mount Diablo, and is actually a Cal State alum from undergrad at San Jose State with a degree in meteorology. Matt and I met during his PhD at University of Arizona, where it looks like he'll be presenting at least some of his data and talk from today. And you know, like me, Matt's kind of a broad thinker, I'd say, wears many hats: eco-hydrologist, ecosystem ecologist, biometeorologist, probably all fit really well. Some fun facts about Matt is I know he's an avid musician, is one of the best ones. And just the kind of my personal take, over the years I've introduced Matt to quite a few people, socially, professionally, whatever, and almost every time it somehow finds its way into the conversation. One of the smartest people I know. So, take it away, Matt.
Thanks, John. Hello, everybody, and thanks for the opportunity to come up here. Can you hear me? I enabled the mic. Is it good? Okay.
I don't. Is it? I don't think it's miking.
No, it is. It's just not very loud.
Okay.
Just up both sliders. Why not?
All right, let's try that. Okay. Any different? I can talk louder too if that's helpful. Okay. If it becomes a problem, let me know. So yeah, this is it's cool to be here. As John said, I grew up in California, and I haven't been to Chico in a very long time, so it's nice to see it. And today, this is an image of a field site in southern Arizona where I did a lot of this research and where I got to know John, working with this eddy covariance instrumentation, which really allows us to understand the breathing of the biosphere and how these ecosystems are responding to different management and climate change factors. An outline for today, I'll start off with, why do we want to study drylands, what's interesting about them, some background on changing water availability, and then 3 projects in the dryland research area. And then at the end, I'll talk about some new research I have in these more agricultural systems and some similarities between drylands and irrigated agriculture. So as we all are well aware, as the world warms, that changes our water availability. Let's see if I can get there we go. So this is from 2021, and we had a large parts of the western U.S. in this severe drought. And that picture looks different today thanks to the wet winter that we've had, but by no means is this gone. And we're going to see this return in the future. That's kind of a given. And when we look ahead to the future, this is a map of what the model sink soil moisture will look like. You can see these brown colors just indicating projected losses in soil moisture. And that's due to 2 main factors. Really, this rising atmospheric thirst. The atmosphere is aggressively evaporating water from the landscape. And also, we're observing changes in our water supply. So rainfall is tending to become more intense. We're seeing these fewer but larger storms that are separated by longer dry periods. And this is important because soil moisture is really a key driver of how these ecosystems function and how healthy they are. Drylands are interesting because they cover large portions of the globe. They're home to billions of people. And we define them as places where the amount of energy to evaporate water exceeds how much water is provided in precipitation. And this imbalance makes them sensitive to changes in climate that change rainfall patterns. And they also provide a lot of ecosystem services. So things like food, fuel, and fiber. They also help regulate the climate by taking CO2 out of the atmosphere when the plants are healthy. And they also help conserve soil resources and play key roles in nutrient cycling. And so these are important places to study. Additionally, they're important in terms of the global carbon budget. So when we look at a time series of how the land carbon sink is changing, this is if we were to add up all of the ecosystems on land and how much carbon they're taking out of the atmosphere, you can see that over time there's a lot of wiggles in this. There's a lot of noise, there's a lot of variability. And research has shown that a lot of that variability is driven by these semi-arid ecosystems. And that's because they're really sensitive to changes in moisture. If it's a wet year, they might have a lot of productivity, they might take a lot of carbon out of the atmosphere. But if it's a dry year, they tend to kind of shut down. And that turns out to drive a lot of this year-to-year change in the land carbon sink. And they're really controlled by water availability. So this is an image of a grassland in Arizona, and you can see that it's not really happy, right? This isn't the dry season, the plants aren't really happy. But when the monsoon comes, we get these rainfall patterns that really green up the grasses. We have a lot more moisture in the atmosphere and soil moisture, so there's a lot more happening once the water gets in the system. And so kind of, because they're so dependent on moisture, they're really interesting places to study. And when we think about water availability, this is a recent paper that Russ Scott and myself and John contributed to, where we're looking at the net ecosystems production of carbon dioxide, so where positive values indicate carbon sink, carbon uptake, and negative values by a carbon source. You can see that these lines are really dependent on how much precipitation the ecosystem receives. So we really need a certain amount of precipitation to be above this, into this positive range to help these ecosystems take carbon out of the atmosphere. And so my kind of overarching question here is, well, how are these changing rainfall patterns and water availability, how does that uptake or change our carbon balance in drylands? And to do that, I use this method called eddy covariance that I'm sure you've heard about before. If you can think about how wind moves across a landscape, as it flows over the ground, there are all these obstacles like trees and shrubs and various things. And those generate these mechanical eddies. And it turns out that these eddies are really good at mixing things in the atmosphere, really good at transporting things from the Earth's surface into the atmosphere. And if we monitor these eddies with a measurement platform, we can measure the exchange of things like carbon dioxide, water vapor, heat, and other things like methane and other gases. And so we really need two pieces of information here. We need to know the concentration of the gas, which is provided by an infrared gas analyzer. And we need to know the wind, and we get that from a sonic anemometer. And with these two pieces of information, we can compute the product that gives us the flux, which is the exchange of either water vapor or carbon dioxide. And so these are powerful tools for understanding ecosystem function. If we're interested in water exchange, we're directly measuring the evapotranspiration that's coming off the landscape. We're also at the same time measuring the net exchange of carbon dioxide. And we can break that down into the carbon that's coming in through plant growth, photosynthesis, and the carbon that's coming off through respiration in the soils and plant maintenance. So the first project I want to go into more detail about is using these eddy covariance towers to look at the role of atmospheric aridity during rain pulses. In these ecosystems like the ones we studied in southern Arizona, we have these kind of episodic rainfall events where part of the landscape might be receiving rainstorm where the other parts of the landscape are still dry. So there's kind of this flashy nature of it rains and then it's dry and then it rains and then it's dry again. And if we think about this in terms of like a conceptual framework, we might draw a pattern of soil moisture like this, where it's pretty dry, but rainfall boosts our soil moisture. And as the time progresses since the rain event, we start to dry down back into this kind of water stress period. And so we might expect the ecosystem to respond like this, where we'd see a pulse of our photosynthesis and a pulse of evapotranspiration, both of which tend to decline as the soil moisture becomes limited. And this has been a very useful framework for thinking about how these ecosystems work. However, there's really more going on, and as we continue to pump greenhouse gases into the atmosphere is becoming more thirsty, and it's evaporating water out of soil and out of plants more aggressively, especially during the spring and the summer in the western U.S. And we call this vapor pressure deficit. And plants are smart, right? When the vapor pressure deficit is high, when it's really dry out, they close their stomata. These are the valves that gases flow through to let plants grow. And they want to save water. But the problem is, by saving water, that also shuts down their growth and their productivity. And so what our colleagues have found is, when we look at photosynthesis for a variety of different ecosystem types, shown as the symbols, as the air gets drier through higher VPD, we see this reduction in the photosynthesis at the ecosystem scale. And so what I wanted to do here was to apply this thinking to these rainfall events. Basically taking the hypothesis that, we might see a normal situation in the solid line, but if the atmosphere is dry and we have high VPD, we might expect something like this, where we have a big suppression of our photosynthesis during the course of that rain dry down event. So to test this, I looked at a bunch of rainfall events from 4 sites in southern Arizona that spanned different kind of structures. So this is like a shrubland with patches of soil, bare soil in between. This is a savanna where there are mesquite trees and some seasonal grass growth, and 2 kind of more semi-arid grassland type systems. And what we found is that when the VPD was high, so when the atmosphere was really dry, the dashed line here, showing a reduction in the photosynthesis of the ecosystem throughout the entire pulse event about 10 days after the rainfall. So this evidence that when the atmosphere is dry, we tend to lose some of that productivity. In terms of our evapotranspiration, when we average across the event, we don't really see a big difference between the normal and the dry atmosphere conditions. But we might see some indications toward the end of the rain pulse event, where the dashed line is lower, which could indicate that the plant stomata are kind of shutting down, and that's causing the transpiration to reduce in response to that dry air. When we put these 2 pieces together and think about water use efficiency, that's the ratio of photosynthesis divided by the water use. So some people call it like a crop per drop in agricultural systems. Dashed line here, the high VPD situation, we're losing our water use efficiency. So these ecosystems are not getting as much productivity, not as much photosynthesis for how much water they're using when the atmosphere is dry. Respiration, this is the carbon that's being emitted from the landscape. We didn't see a big difference here, and that was not surprising, because we didn't expect there to be any direct link between the air's VPD and the respiration of the ecosystem. In terms of the net carbon uptake, that's the difference between photosynthesis and the respiration. Again, the dashed lines here showing reductions in how much net carbon uptake is happening when the atmosphere is dry. That was just for the savanna system, but when we looked at all the different ecosystem types, including the grasslands and the shrubland, all these tended to behave similarly, in that their photosynthesis and their carbon uptake was reduced when the atmosphere was drier. And so this could mean a loss in the productivity in the future with higher VPD, even if the amount of rainfall doesn't change. So when we kind of think about how are these systems responding to changing water availability, we may expect a reduction in the carbon uptake. But we know that that's not the whole picture, right? We have the water demand side, but we also have the water supply side, that rainfall intensification. And so that was kind of the next part of my research program, is to look at that. So to look at this process, I worked, again, with John at this site in southern Arizona. This is the Santa Rita Experimental Range. And we helped a colleague build these shelters. These are rainout shelters. And they basically block they block rainfall, so no rain gets to the plots that are beneath them. And then we came in and hand irrigated them to control precisely how much water was applied at a different time. And that was to really mimic the climate change that we expect of this transition in rainfall patterns. And we were looking at the soil carbon emissions and how that responded to these changes. So when we think about our ecosystem carbon balance, we're thinking about respiration again. That's the carbon that's lost. And particularly, we're focusing on the soil respiration. So that is the main pathway that carbon is lost from ecosystems, aside from things like fire, disturbance, things like that. And it is the amount of carbon that's coming out of the soil surface that's the sum of microbial respiration. So bacteria and other things in the soil, they eat soil carbon and then they respire CO2 just like humans do. Well, we don't eat soil, but we respire CO2. And there's also root respiration. So the roots of plants, to stay alive, they emit CO2 as part of their process. And so to measure that, we use these automated chambers that basically every hour this chamber head will swivel onto the collar here, make a measurement, and then return to this position so that we're exposing the soil to the ambient conditions, but we're still getting a lot of measurements, and you don't have to go out there every day or every week to get a measurement. And the motivation for this, again, we've got drylands covering lots of places, but when we look at where people have measured soil CO2 efflux, we see like large gaps in some of the drier places, right? So there's not as much coverage in some of these drier areas. And so we looked at a couple different factors here. We looked at the roles of soil moisture and temperature, but also how the plants were shaping this soil carbon emissions by looking at chambers that were near vegetated ground and chambers where we removed all the plants and dug these trenches to prevent the roots from growing back into the plots. And what we found is that the response of the soil respiration, that efflux of carbon from the soil, that was a function of temperature, but it was really dependent on the amount of moisture in the soil. So when we had wet soil, like these green dots, we had this really strong positive exponential increase in the efflux with temperature. But when the soils were dry, we didn't see as clear of a relationship, and that sensitivity was much lower. And this is important because a lot of the understanding we have of soil respiration is really developed in these wetter, more moderate climate places where they expect, like in Harvard Forest in Massachusetts, this more exponential response that looks like what we see when there's moisture. But in drylands, we're often so much in this period of dryness that if we were to model it just based on temperature, we would really be missing the true picture. We also found some interesting differences between the vegetated and the bare chambers. So this is looking at the accumulation of carbon emissions over the season here, and we can see much higher values in the vegetated plots compared to the bare plots. And that's just because we have a lot more roots and root activity contributing to the soil carbon emissions in these plots near grass. And the next step we took here was to try to reflect these processes in a model. So this is what we get when we take the observations. Again, we see kind of this water stress period, but at the onset of the monsoon, a big increase in our soil respiration, and then a decrease during the dry after summer period. If we were to model this just based on temperature, we would get it totally wrong. So we would really overpredict in the dry season, and then in the main growing season here, we would really underpredict. But when we add a moisture term to this model, we really capture that seasonal transition much better. So we're suppressing the respiration again in the dry period and then ramping up in the wet period. But we're still really underpredicting during that kind of main monsoon growing season. And so the next thing we did was to add a term to represent the ecosystem's plant growth, and we really got a better agreement here by considering the photosynthesis of the ecosystem. And So this is just showing that we really need to think about these factors if we want to understand the carbon cycling in these dryland systems that is often much times, in many ways, different than it is in more moderate climates. So we haven't really solved the question yet, but we know that the carbon emissions response to the change in water is probably going to have something to do with these factors. To really pinpoint what's going on, we did one more experiment where we went underneath these chambers, or underneath these shelters, and looked at what happens to these chamber measurements when we're controlling the rainfall. And this was a really, at times, challenging time to work, right? So this is 2020. We were wearing masks. It was the hottest summer ever recorded in Arizona, and we had there's very poor ventilation beneath these. So it was a time to build camaraderie with your fellow field researchers. And the motivation for this is when we look at the observations, our colleagues have found the frequency of rainfall over time is decreasing, that red line showing a decrease in how many rainy days there are per year, and also an increase in how variable that rainfall is. And also, this green line here showing over time, the length of dry days in between rain events is increasing. So we're seeing this transition toward a future with these fewer storms that are separated by longer dry periods. And so, again, that was the motivation. And to answer this question, we returned to these rainout shelters here. Really nice view of the mountains back there. And here's an image of the field technician here, or the postdoc working on this project, Nate, and he's hand irrigating these plots. And he has a flow meter. This is really high tech, right? So he's just measuring how much water is applied very carefully to each of these plots. And what we did, we gave all the plots the same amount of water at the beginning of the year, but after that we changed the size of these storms that we kind of manufactured. So we have this purple regime showing if we water these plots frequently, but with small amounts of water. So, very small amounts, but happening a lot. And the green here showing, we only have a couple of these rainfall events, but they're really big, and they're separated by long droughts. And we made sure that at the end of the season, all these plots got the same amount of total rainfall. So what we're really doing here is like we're repackaging the amount of rainfall into different sized storms. And the chamber data here, this is the accumulation of carbon emissions over time. We found that there were higher emissions in that many small irrigation regime, and there was a reduction in our emissions when we had those few but large irrigations. And the reason to try to figure out why that was, we looked at the relationship between respiration and soil moisture. And we found that the slope of this line was much higher in the many small rainfall than it was in the few large rainfall regimes. And so what this means is that for a given amount of soil moisture, you're going to have a higher amount of carbon emissions happening if that water is applied more frequently in smaller doses. And what we think is happening is the frequent wetting is keeping the microbes maybe more happy or stable, and allowing them to do their thing and respire. Whereas the kind of infrequent rainfall, that's these really long, like several week dry periods in between the waterings. And that might be causing the microbial community to either die or go into some sort of like hibernation mode. And that's not something we're really thinking about in the models. And so when we think about our picture here, again, we've got this atmospheric drying. We're expecting a reduction in how much carbon is sequestered. As rainfall intensifies, we think there could be a reduction in how much carbon is lost. And so that kind of presents a new hypothesis to test, right? Understanding, well, if these are opposing responses, does that kind of buffer the ecosystems? Does the fact that we're losing less carbon kind of buffer the fact that we're also gaining less carbon? And I don't really have a good answer for that yet. I've kind of changed research directions with my new position, but if anyone is interested in that, I'd love to talk about it. and think about it more. We may have some insights into what's happening here when we look at these really long-term records. So Russ Scott in Arizona has been running these flux towers for like 24 years, I think. And that's a really long data set to really see what's happening. And what he's showing is that when we look at photosynthesis here and respiration here, this is moisture on the x-axis, the slope of the photosynthesis tends to be higher than the respiration. And so that could mean that a reduction in your moisture could have a stronger impact on photosynthesis than it might on respiration. So I wanted to kind of show some examples, moving on from the dryland stuff to ways that dryland results might apply in these kind of dryland agricultural systems. So in the Central Valley of California, as everyone who lives here is aware, it gets really hot and dry in the summer, and basically becomes this water-limited situation. And so I'm using these the flux instrumentation and the soil chambers to look at these processes in agricultural systems. And this is an example of how a vineyard, the irrigated vineyard, responds to that BPD. Again, that's that atmospheric drying parameter. So this is evapotranspiration. That's how much water is being evaporated from the landscape. And the x-axis here is the BPD, that atmospheric dryness. And what we see is that when BPD is relatively low, there's a positive relationship. So higher BPD gives you more evapotranspiration. And that makes sense. There's more energy to evaporate water, to help the plants use water to transpire. But there seems to be a sweet spot here at which higher, as we continue to increase, start to see these reductions in evapotranspiration. And what could be happening here is that the BPD is getting so high that the plant stomata are closing. They're no longer transpiring water because they're just shutting down. And so this could have, implications for an irrigation manager who's trying to figure out how much water to apply to their plants in the context of a changing atmospheric dryness. When we look at the photosynthesis, again, we see losses in productivity as BPD increases. Each line here is a different vineyard. These are different vineyards in different regions of California. And what I find interesting is, kind of with the exception of maybe one or two of these sites, these have kind of a similar slope, and that could indicate they have a similar sensitivity in their BPD response, even though they're different varieties and different places and different growing practices. When we look at the water use efficiency, this is the ratio of photosynthesis to ET. You can see this pronounced reduction here as we get to higher BPD. So that's showing that as the BPD rises, we really are using water less efficiently. We're getting less crop growth for the amount of water that is being applied. And I think that's all I want to show about this picture. So it's not all bad news. I think that there's potential for carbon sequestration if ecosystems are managed intentionally and in a more strategic way. So here in the Mediterranean climate, we've got lots of sunlight, we've got lots of warm temperatures. Those are what plants need to grow. So we have a lot of potential for plant growth, a lot of potential for photosynthesis. And we also, if we think about crops like vineyards or orchards, they're perennial. So there's a lot of biomass here that's accumulating above ground, and also the rooting systems that we don't harvest each year. We don't go out and cut that down, and it stays on the landscape. And cover crops are another way that represents an additional way to sequester carbon in these ecosystems. We also have things like mowing and pruning that can change how much carbon is being moving through these systems. Often they're drip irrigated, which causes these gradients in moisture where we have a lot of moisture near the drip lines for the trees, and then drier patches in the middle. And so there's a lot of things going on that we need to think about in these systems. And that's what we're doing. So when we look at the net carbon flux, positive values here are carbon source, negative values is carbon uptake. You can see that as we get into the spring here, we transition into this carbon sink. activity. And that's because we've got mostly this cover crop here that's growing aggressively, it's photosynthesizing, it's pulling carbon out of the atmosphere. And the vines are actually dormant at this period. We have periods when we mow here. You can see the system transitions pretty immediately into a carbon source. So we lose all that biomass. There's no more photosynthesis happening. And instead we have emission of carbon. But pretty quickly we recover our cover crop biomass, and then the ecosystem becomes a sink again. During the main growing season, this is when the canopy is nice and green and fully expanded, but the cover crop is dry. This is when we get really high values of carbon uptake. And those kind of taper off as we start to see the canopy senesce. And this is interesting. When we till the ground, which is when we disturb the soil, cultivate it, that causes a big pulse of your carbon emissions. And so there could be ways of reducing this carbon emission by avoiding tilling or using less frequent tilling. We're doing some experiments to better understand cover crops and how they impact our carbon and water balance. We did a side-by-side comparison where we looked at vineyards with cover compared to vineyards with bare alley space. And we found some kind of interesting results. So initially in the springtime, we found that the cover had negative values here indicate more uptake of carbon compared to the bare soil. And what happened here was that we had more photosynthesis happening in the cover crops than we did with the bare ground because there's no vegetation there. And so we thought, oh good, that's going to result in more carbon uptake at the end of the year. But by the end of the year, it was actually difficult to distinguish if there was any significant difference between these two managements. Yeah. And what we think happened is we actually had higher respiration values in the fall for that cover block, because there was way more biomass that was incorporated into the ground. And when they came and used the tractors to till the soil, that just enabled all of that biomass to then be respired and lost back to the atmosphere. And so I think this shows that there's these key links right between our photosynthesis, our growth, and our decomposition. And if we're going to really have meaningful carbon sequestration, we're going to need to do more than just cover crops. We're probably going to need to do cover crops in combination with other regenerative practices, like reducing our tilling or avoiding tilling entirely, or else we're just going to delay when that carbon is returned to the atmosphere. And one of the big concerns about cover crops is that they use water, and water is king in California. But I think growers they're smart. They know how to manage these ecosystems to avoid water competition. So what we did here, we had these cameras that were taking an image every day of the system, of the ecosystem, and I drew boxes around the vines here in red and around the grass here, and just extracted the greenness trends over time. So this is a time series of greenness. You can see there's no greenness in the vines in the winter. That shoots up in the summertime. And that's totally the opposite of what happens with the cover, right? So cover is green in the winter, and it totally decreases in the summertime. So these are kind of opposing patterns, which kind of shows that growers can manage these things to avoid direct competition for water resources, while maximizing how much photosynthesis the ecosystem as a whole accomplishes. Another thing we're doing is we're looking at cover crops in not just vineyards but in orchards, and we have this really interesting side-by-side comparison here. We have two blocks of a young orchard. One, we've removed all of the cover, and in the other, we've retained the cover crop. This is an image showing the difference here. And we've measured, we have 2 flux towers, one in each of these sites. We also have our soil chambers in these locations to try to really understand what's going on. This is some pretty new data showing the flux tower data. So this is latent heat flux, or evapotranspiration, the average daily pattern. For the cover crop plot, we see higher values of evapotranspiration, so more water is being used. But also we see more carbon uptake is happening. And it's kind of interesting that the difference is bigger for the carbon than it is for the water. And so I think this is something we're interested in monitoring, not just for the springtime, but for the whole year, and to see how this teases out over multiple seasons. We're also looking at the soil, water, and cover crop question using these devices that measure soil moisture. They kind of look like a giant drill bit that you screw into the soil. And along the side of them, they have these moisture probes along the whole depth. And so we can get a really good picture deep in this, as we go from the surface down to about a meter, how the moisture is changing over time. And so we have some of these in our no cover block, in our cover crop block, and I don't know how to interpret these data yet. This is like 3 days old data, but it's going to be really interesting to see how, at different depths in the soil, how the moisture compares. Like, are the cover crops using moisture in the surface, but helping get better infiltration of water deeper below? How does that change seasonally? Different years, maybe a wet year versus a dry year. So these are kind of ongoing questions for our group. And this is important because, like, in a year like we just had, right, we had these atmospheric rivers. And just visually, we don't even need data to see the difference here. When we have cover crops, we don't see as much water ponding on the soil. When we don't have cover crops, we see a lot of water ponding, meaning there's a potential for the water to be evaporated, there's a potential for erosion of our soil resource. And so this will be interesting to monitor as we move on. This is the last kind of stuff I'll present today. Again, we have these automated soil chambers, and we have them deployed in the orchard. So in one orchard, we've got that cover crop, in the other orchards, we don't. And we're looking at how those respiration rates change in the drip line and how they compare in the mid-row for different ground cover management. And what we're finding, again, is that when we have the vegetation growing next to the chambers, we see much higher values of that soil respiration, that carbon coming out of the soil. So compared to the more, just bare ground situation. And so that could indicate that there's more soil health, there's more microbial activity, there's more carbon being cycled in the soil when we have these grasses growing, the cover crops growing. And this is similar to a plot I showed earlier. When we look at the response of that respiration to temperature, this is in the bare soil, right? We don't see a clear relationship here. But when there's vegetation, we see this more exponential increase in our respiration with temperature. And we notice the interesting effect of the time of day. So if we just focus on, like, say, 15 degrees of soil temperature, you can see that for 15 degrees, the respiration could either be like, at the top of the range 6 down to like 4. So a big range, and that was really explained by the time of day. So if you look at these light-colored blue, that's happening in the midday period when we have the plants growing, compared to these pink dots. This is in the evening time. So what we think is happening is that in the daytime, the photosynthesis of the grass is putting carbon into the soil that the microbes are then using to respire right back to the atmosphere. So there's more cycling happening. And so that's what I have to share today. I just want to acknowledge co-authors and colleagues here and leave you with a nice picture. And I'd be happy to have some discussion or questions or anything you might have. So thank you.